Welcome to my presentation titled In-Silico CLIP-seq, Predicting Protein-RNA Interaction via Sequence-to-Signal Learning. In this presentation, I'm going to present to you a method to predict protein-RNA interaction at nucleotide resolution from the RNA sequence. RNA-binding proteins are crucial actors of post-transcriptional regulation, and while there are many methods to measure protein-RNA interaction in vivo, one of the most prevalent ones nowadays is eCLIP, which stands for Enhanced Cross-Linking Immunoprecipitation. Of course, the availability of measuring protein-RNA interaction is subject to the availability of transcripts in vivo. So in order to measure or prevent repeated experiments to measure the protein-RNA interactions between all possible transcripts, a popular approach is to use computational methods to predict RBP binding to an RNA of interest from the RNA sequence. Computational methods can also help to identify binding preferences of target proteins, and they can be used to score the impact of sequence variants on protein-RNA interaction. Commonly, computational methods consist of machine learning methods which are trained to predict whether a given RNA sequence is bound or not bound by the protein of interest. So a common workflow would be to use existing eCLIP or other CLIP data for a protein of interest, identify regions from a transcriptome that are bound or not bound, and then use those binary labels to train a machine learning binary classifier. Classification-based methods, however, have several limitations. First and foremost, the predictions have relatively low nucleotide resolution. So that means that the predicted label is assigned to the entire input RNA sequence, which can be hundreds of nucleotides in length. But sometimes we might be interested in more fine-grained predictions, so maybe for a few nucleotides or even at nucleotide resolution. The other issue is that these methods heavily rely on pre-processing for labeling. So one needs to first employ some peak calling algorithm to map the experimental data for each region into binding, non-binding. And this is of course associated with loss of information. So we sometimes have quite complex experimental signal footprints, and those get basically mapped to a single bit, that is binding or non-binding, which is associated with loss of information. So how can we model protein-RNA interaction, or rather cross-linking between proteins and RNA at nucleotide resolution? We can maybe leverage the fact that iCLIP or eCLIP and other nucleotide-wise protocols generate nucleotide-wise data, and usually in the form of cross-link counts. And we could attempt to predict the cross-link counts at each position of an RNA directly. However, this is pretty difficult because the absolute number of observed cross-link counts is heavily dependent on the availability or the abundance of the RNA at the time of the experiment. That means that RNAs that are highly expressed will automatically have a higher number of observed counts, and the number of counts does not directly translate to the binding affinity, and more often to just the abundance of the RNA. Of course, this is controlled by different processes, mostly transcriptional processes that are not encoded in the RNA sequence. So a more viable approach is to, instead of predicting the counts directly, to instead model the distribution of counts along the sequence. And the distribution of counts we can expect to be subject to the underlying RNA sequence. So the central idea is that given an RNA sequence, and assuming that there are some cross-linking counts that fall within this RNA sequence, our goal is to predict a probability vector of the same length as the sequence, which indicates how counts are distributed across each nucleotide. So we assume that counts are coming from a multinomial distribution. Therefore, the probability at position I, so the predicted probability pi, corresponds to the fraction of counts that this position will receive among the total counts. Now, if we want to train a machine learning model, we somehow have to evaluate the goodness of our prediction, and we can do that by using the observed count data. So given our prediction and the observed count data, we can compute the likelihood that the observed or that the predicted probability vector p generated the observed distribution of counts. And the observed distribution, of course, comes from the actual experiment. Then, by computing the negative log of this likelihood, we basically computed a loss, which we then can use to train a model via gradient descent. The issue with CLIP, so eCLIP as well, is that it's subject to technical biases. For example, libraries, or like often when one targets specific person of interest of an antibody, there's often co-purification, so that the target library is enriched with crosslinks of other ABPs. Also, some nucleotides are more photoreactive than others, so several biases are introduced during the eCLIP protocol. Fortunately, eCLIP is usually paired with a control experiment, the size-matched input control, which basically omits the immunoprecipitation step, so it represents a kind of broad background binding signal. So how do we use this control sample to correct for CLIP-seq biases? First, just like we modeled the CLIP crosslink counts, we can also model the crosslink counts of the control from sequence. And we then reformulate how we model the total, that is the eCLIP crosslink counts. So, given our prediction of the control crosslink count distribution, as well as the prediction of an unobserved, quote-unquote, true crosslink count distribution, which represents the crosslinks of our target protein, we define or redefine the crosslink count distribution of the CLIP as an relative mixture between the target and the control predictions. And we control the relative intensity of the bias or the control sample using mixing coefficient pi. So the lower the mixing coefficient pi, the less influence the control track has on the total counts. So putting it together, it basically a general, we can model the distribution of crosslink counts in the following step. First, we tile the transcriptome into fixed length sequences. We then predict the distribution of crosslink and hidden protein-specific counts via a ResNet. So we use an RNA sequence as input, and the outputs are two probability vectors, one for the control experiment and one for the hidden target experiment. We then predict the mixing coefficient from sequence and mix those distributions, the control and the target, additively. And this then gives the distribution of the final CLIP/eCLIP. Then given the true counts of the eCLIP, so the total, as well as the control experiment, so in the case of eCLIP, the size-matched input, we then compute a negative log likelihood loss, and we proceed to train a model using gradient descent. So here's an example prediction. At the very top, these sticks, you can see the true counts in blue for the eCLIP and in red for the control experiment. And then below you can see the predicted distribution of crosslinks between the eCLIP, control and target respectively. So as we can see, the predicted the modes of the predicted distribution of the eCLIP and the target correspond to the modes of the actual counts. And the top mode is also associated with the known canonical binding motif of the QKI protein, for which this prediction is. On the other hand, for the control prediction in red, we can see that the modes rather correspond to urate regions, which are likely contaminations with a new binding RBP. So one nice aspect of our model, which we termed RBP-net, is that since it's purely convolutional, it can basically operate on sequences of arbitrary length. That means that we can, at inference time, input whole transcripts up to tens or even hundreds of thousands of positions and obtain a prediction for the transcript in a single forward pass. So here's just an example. In blue, the true counts, and in yellow, the predicted distribution. And crucially, you can also see in red the single nucleotide peaks that were called with a single nucleotide peak caller called pure CLIP. And by treating the positions that are residing within peaks as positives, so regarding the called peaks as ground truths, and everything else as negatives, we can associate a performance score, in this case the area under the rock, for each transcript, which we then can average across all transcripts to gain a experiment-wide performance metric. We did this for 70 different eClip experiments, and we compared the performance of our method, RBP-net, versus a state-of-the-art classification-based approach called DeepRipe. And we can see that our performance is better on every single eClip experiment. So once we perform the prediction, we can also query the model to explain which positions in the input sequence were particularly guiding this prediction. And when we perform this at large scale, we can start to analyze important subsequences or nucleotides to identify what are the binding preferences of our protein. So this is exactly what we did. We performed predictions over thousands of sequences and then computed feature attribution, feature importance scores. We extracted highly important 5-mers from our feature importance maps, and we then aligned those important 5-mers greedily, starting from the most important 5-mer, in order to form consensus motifs. And as you can see here on the very right, the consensus motifs we built correspond to literature motifs, for instance for the QKI, RBFOX2 and TRI1 protein. So, like I said, we compared our build motifs to literature, and what was interesting on the left-hand side is that when we use feature importance scores computed on the target track, so the unobserved protein-specific prediction track, we saw a much higher agreement to literature motifs than when we used the total track, which still contains some degree of bias. Also, on the right-hand side, we can see that the top 20 highest important 5-mers obtained from the RBP-net target track is more or less on par with Pika, which is a dedicated motif discovery tool, and it's higher than RBP-net, DeepRipe, and especially the RBP-net control prediction, which we expect to mostly capture bias. So another application of RBP-net is the scoring of impact of mutations on protein-RNA interaction. So for classification-based method, that's pretty straightforward. Usually you predict on the reference allele as well as the alternative allele, and then the impact score is more or less the difference between the predicted binding probability between the reference and the alternative sequence. For RBP-net, that's a bit more difficult because our predictions are vector valued. So we have to compare two different vectors, prediction vectors with each other. So in this case, you can see that we have predictions for the reference allele in blue, which has 2 very stark modes. And when we introduce C2a mutation, the predicted distribution of crosslink counts changes quite a bit. First of all, the modes drop significantly, and the probability mass then gets reallocated to other positions, in this case more upstream around position 50. So in order to score the impact of mutations, we can basically quantify this change in the predicted probability mass by comparing the predictions of the reference and alternative alleles via the KL divergence, which then serves as an impact score. And you can see here on the right hand side, if we introduce mutations within this canonical motif, we gain much higher impact scores than if we mutate outside the motif. So one application of the variant impact scoring we checked out is that we wonder whether we can identify splicing associated variants via impact scoring of splicing associated IBPs. So basically the idea is that we use IBPs that are associated in splicing, and we score the impact of variants and check whether the impact scores can then differentiate between splicing associated and background mutations. So for this task, we selected 232 splicing associated variants as well as background variants from GNOMAD, and we performed impact scoring with splicing associated IBPs. And as you can see, indeed, unpredicted impact scores can very well separate splicing associated variants from background variants for a number of different splicing associated IBPs. So in conclusion, RBPNet can model protein-RNA interaction at nucleotide resolution. It operates directly on the raw count data and does not depend on p-calling for training. It can predict on variable length sequences as well as whole transcripts. And it can identify binding motifs via feature important scoring, as well as it can score a variant impact, the impact of sequence variant on protein-RNA interaction. If you're interested in more, please attend my talk, as well as check out the poster at 1325, or just approach me at the conference. Thank you very much, and I hope to see you in person in Lyon.